A very good evening to all. Today we are going to discuss biomolecules. We'll do a quick revision of biomolecules. So let's start with the lecture. Before we proceed with the lecture, let's take a pre-reading question, and the question says match the list 1 and the list 2. List 1 contain protein, unsaturated fatty acid, nucleic acid and polysaccharide. List 2 contain C double bond, phosphodiester bond, glycosidic bond and peptide bond. Choose the correct answer from the option given below. So you have to match the list 1 with the list 2. We know that protein is a polymer, the macromolecule, and the monomeric unit of protein are amino acid. B says unsaturated fatty acid. Fatty acid, they are the long chain of hydrocarbon. They have long chain of hydrocarbon and one carboxylic group. Fatty acid, there are two types, saturated fatty acid and unsaturated fatty acid. Saturated fatty acid, they have a single bond, and the unsaturated fatty acid, they have double bond. It could be 1 or 2. Third is nucleic acid. Nucleic acid is also a polymer, and the monomeric unit of nucleic acid are nucleotide. D says polypeptide. So D says polysaccharides. When many monosaccharides they join, they form a polysaccharide, and polysaccharide is a carbohydrate. Now let's match the list 1 with list 2. A says protein. We know that the monomeric unit of protein are amino acid, and two amino acid, they are linked to each other by a peptide bond. So A goes with peptide bond, A fourth. B, unsaturated fatty acid. I told you that fatty acid, there are of two types, saturated and unsaturated fatty acid. And unsaturated fatty acid, they have double bond. So B, 1. C is nucleic acid. We know that the monomeric unit of nucleic acids are nucleotide. And nucleotide, they consist of phosphate, sugar, and nitrogenous bases. The bond between the phosphate and the sugar is the phosphate ester bond. And the bond which link the two nucleotide is a phosphodiester bond. So C match with phosphodiester bond, so C2. D is a polysaccharide. We know that the monosaccharide unit, they join together and they form a polysaccharide. And these polysaccharides, they have a linkage which is known as a glycosidic linkage or the glycosidic bond. So D match with 3. So A fourth, B1, C2, and D3. A fourth, B1, C2, and D3. So correct option is option number 1. I hope it is clear to all. Now let's discuss with biomolecules. Bio means living. So all the molecules which are present in a living organism are known as a biomolecule. So two analysis has been proposed by the scientists. Number 1 is the chemical analysis, and the second one is the ash analysis. So in the chemical analysis, what the scientists did, they take the living tissue, they grind it with the trichloroacetic acid, and they make a slurry. Now they use a funnel tube. This is a funnel, this is a beaker. Now they pour the slurry into the funnel and they use a cotton for filtering to filter the slurry. So when they pour the slurry into the funnel, the filtrate, this beaker, contains the filtrate. And this filtrate contains the acid-soluble pool. Why acid-soluble pool? Because we grind this living tissue in the trichloroacetic acid. We use this chemical trichloroacetic acid. That is why we are getting the acid-soluble pool. Soluble, the molecules which are smaller in size, they easily get dissolved in the acid, and so that we are obtaining these micromolecules. We call these molecular micromolecules. So we are obtaining the micromolecule in an acid-soluble pool. And the part which written in the top is known as retinate. And this retinate, it consists of acid-insoluble pool. Acid-insoluble pool, it consists of macromolecules. Second analysis is the ash analysis. In the ash analysis, what the scientists did, they take the living tissue, they dry it, and they burnt it. So the carbon is removed in the form of carbon dioxide, and the ash, which remain, has the inorganic element like magnesium and calcium. Now let's discuss the metabolites. There are two types of metabolites, primary metabolites and secondary metabolites. Primary metabolites are those metabolites which play a very important role in the normal physiological process of the body. Like sugar, protein, these are the examples of primary metabolites. Secondary metabolites are those metabolites which are not essential for the normal physiological process. Like rubber, carotenoids, alkaloids, essential oil, toxin, polymeric substances, they all are the example of secondary metabolites. Secondary metabolites, they act as a byproduct. This table is given in NCERT, and this is a very important table. Many times the question came from this table. Next, we are going to discuss protein. We know that the two amino acids, they are joined by a bond, and the bond is known as a peptide bond. And the monomeric unit of a protein is amino acid. So when many amino acids, they join together by the peptide bond, they form a long chain, and that long chain is known as a polypeptide chain, which is referred as a protein. This image is showing the structure of amino acid. This amino group is present, the carboxylic group is present. This side chain R is present, and this hydrogen. This is the normal structure of the amino acid. And depending upon the number of carboxylic group and the amino acid could be acidic, basic, neutral. Acidic, if they're having a more carboxylic group as compared to amino group, then the amino acid are said to be acidic. Example, glutamic acid. If the amino group are more as compared to carboxylic group, then the amino acid are said to be basic in nature. Example, lysine. Neutral, when there's an equal number of carboxylic group and the amino group. Example of neutral amino acid is valine. Their aromatic amino acids are also present, which have a ring-like structure. Like example, phenylalanine and tryptophan. Their essential amino acids are there and non-essential amino acids are there. Essential amino acids are those which cannot be synthesized in our body. We have to take these amino acids from our outside, for example, by eating food. Non-essential amino acids are those amino acids which can easily be synthesized in our body. Collagen is a type of a protein, and this protein is abundant in the animal world. Remember this. If the question asks you like which protein is abundant in the animal world, then the answer would be collagen. But if the question asks you like which protein is abundant in the entire biosphere, then the answer would be rubisco, because rubisco is the most abundant protein in the biosphere, and collagen is the most abundant protein in the animal world. Zwitter ion, when the amino acid, they exist as a dipolar ion, that is, they're having a positive charge and the negative charged when dissolved in water. Proteins, they have four types of structure: their primary structure, secondary structure, tertiary structure, and quaternary structure. Primary structure shows you the sequence of amino acid. They consist of like a polypeptide chain. This shows you the sequence of amino acids. Secondary structure, it has the alpha helix structure, helical structure, and the beta pleated sheath. Tertiary structure, when the protein, they have a three-dimensional structure, and the tertiary structure is very important for the biological activity of the enzyme. Quaternary structure, when more than one polypeptides are present as a subunit, like hemoglobin. Hemoglobin is a quaternary structure. It had two alpha chain and two beta chain. This table shows you the function of the protein. That collagen is a protein and its function is the intercellular ground substance. Trypsin is a protein and it functions as an enzyme. Insulin is a protein. It functions as a hormone. Remember, insulin is a peptide hormone. Antibody is a protein and its function is to fight against infection. Receptor is a protein like sensory reception, smell, taste, hormone, etc. GLUT4 is also a protein and its function is to allow the glucose transport into the cell. Next, we are going to discuss carbohydrates. Carbohydrates, they are the polyhydroxy aldose or ketones. There are major four classes of carbohydrate. There are monosaccharides, the disaccharide, oligosaccharide, or polysaccharide. Monosaccharide when there's present or when there's only one unit is present, like glucose and fructose. They are the example of monosaccharide. Disaccharide when two units are present. Like sucrose is the example of disaccharide. Sucrose is consists of two units, glucose and fructose. Lactose is also a disaccharide. It consists of two units, which is glucose and galactose. Maltose is also a disaccharide. It consists of two units of glucose. Oligosaccharide example is raffinose. Polysaccharide, when many monosaccharide units, they join and they form a polysaccharide, which is a polymer. Example of polysaccharides are cellulose, starch, and glycogen. Remember that cellulose is unbranched and it has beta 1,4-glycosidic linkage. Starch is consist of two parts. One is amylose, other one is amylopectin. Amylose is unbranched. It has one alpha 1,4-glycosidic linkage. Amylopectin is branched. It has alpha 1,4-glycosidic linkage plus alpha 1,6-glycosidic linkage. Please remember this is important that starch consists of amylose and amylopectin, and amylose is unbranched and consists of alpha 1,4-glycosidic linkage. Amylopectin is branched, it consists of both alpha 1,4-glycosidic linkage and alpha 1,6-glycosidic linkage. Next is glycogen. Glycogen is branched. It also has both alpha 1,4 and alpha 1,6 glycosidic linkage. Inulin is a polymer of fructose. Remember this. The starch, they have a helical structure. So because they have a helical structure, so when we treat the starch with iodine, the iodine, they get trapped in this helical structure and cannot move out. Because of which the starch, they give the positive iodine test or the blue-black color. We know that glycogen is branched and the glycogen is the reserve food material in animals. The glycogen, the left end is the non-reducing end and the right end is the reducing end. Reducing means when there's a presence of a free reactive group at the end. Non-reducing means when the reactive group is not free or not present. Remember that sucrose is a non-reducing sugar. Moving next is nucleic acid. There are two types of nucleic acid, DNA and RNA. We know that the monomeric unit of nucleic acid are nucleotide. Nucleotide, it consists of three components, phosphate, sugar, and nitrogenous bases. And if sugar and nitrogenous bases are present, then it is known as nucleoside. There are two types of nitrogenous bases are present, purine, which are double ring-like structure, and pyrimidine, which are single ring-like structure. Example of purine are adenine and guanine. Example of pyrimidine are cytosine, thymine, and uracil. This is a sugar. These are the nitrogenous bases, where adenine is paired with thymine by the 2-hydrogen bond, and the guanine is paired with cytosine with the 3-hydrogen bond. So the nitrogenous bases, they are connected to each other by the hydrogen bond. And the bond between the sugar and the nitrogenous bases is the glycosidic bond. And the bond between the sugar and the phosphate are the phosphoester bond. This is also a phosphoester bond. This is a phosphoester bond, and we collectively call it as phosphodiester bond. This phosphodiester bond, it joins the two nucleotide. The DNA, they have a deoxyribose sugar. And RNA, they have ribose sugar. In DNA, we call it the sugar is deoxyribose. Why it is deoxy? Because on the second position, there is a hydrogen is present instead of hydroxyl group. Because in ribose sugar, which is present in RNA, at the second position, in the ribose sugar, this hydroxyl group is present. But in DNA, the sugar is deoxyribose because on the second position of the sugar, there's a hydrogen is present instead of hydroxyl group. Next, we are going to discuss is lipid. Lipid, they are water insoluble. And remember that lipid is not a polymer. The simplest form of lipid is fatty acid, and fatty acid there consists of long chain of hydrocarbon plus one carboxylic group. You can say this, there's a one carboxylic group, and this is a long chain of hydrocarbons. Fatty acid, there are of two types. There's saturated fats fatty acid or the unsaturated fatty acid. Saturated fatty acids are those which have a single bond. And the unsaturated fatty acids are those which have a one or two double bond. And fatty acids, they are amphipathic in nature. The major example of lipid is a phospholipid, which is found in the cell membrane, because we know that the cell membrane is a phospholipid, it's made up of phospholipid bilayer. This is a polar head, and this is a non-polar tail. Because it had both the group, the polar and non-polar, we call it as amphipathic in nature. And gingerly oil has a low melting point. Remember this. Now let's discuss the enzyme. We know that almost all enzymes are protein, but there are some enzymes But there are some nucleic acids which behave as enzymes, and the example are ribozymes. Remember this example. That all the enzymes, almost all enzymes are protein, but some amino acid, they behave as enzyme, and the example is ribozyme. Enzyme, they are the organic catalyst. There are two types of catalyst, organic catalyst and inorganic catalyst. So inorganic catalyst, they work efficiently at high temperature and high pressure. But the enzymes, they does not work efficiently at high temperature and high pressure because most of the enzymes are proteins. So at high temperature and high pressure, these proteins get denatured. So the enzyme, they does not work efficiently at high temperature and at high pressure. But if we extract the enzyme from the organism which is surviving in a very high temperature condition. So the enzyme which is isolated from the organism which is surviving in a very high temperature condition, that enzyme can work efficiently at high temperature because those enzymes would be thermostable enzyme. For example, Taq polymerase. Taq polymerase, this enzyme is used in PCR, that is polymerase chain reaction, and it is a thermostable enzyme because it is obtained from an organism named is Thermus aquaticus. And the enzyme, they have the active site, the site where the substrate binds. Each enzyme has a specific active site. That means the specific substrate can bind to a specific enzyme. So the enzyme, they show specificity. When the substrate bind to the enzyme, they form a complex which is known as the enzyme substrate complex. And this enzyme substrate complex it's formed for a very short duration. Now, immediately this substrate is converted into the product and the enzyme is released. Now the enzyme, they have a free active site so that it can bind to the next substrate. Metabolism is the sum total of all the chemical reaction that is taking place in our body. So metabolism, they are of two types, the catabolism and the anabolism. Catabolism, when the larger molecule splits into the smaller molecule, because the larger molecule will be break down, so it will release the energy. So during catabolism, energy is released. Anabolism, when the smaller subunit, they join and they form a large molecule. So here the synthesis of a large molecule will occur. So this process requires energy and the metabolism is known as an anabolism. As we know that the reactions, they are catalyzed by the enzyme. So enzyme function is to decrease the activation energy and increase the rate of the reaction. You can see in this image, when the enzyme is not used, the activation energy is high. But when the enzyme is used, the activation energy is lower. So that the rate of the reaction increased. So the enzyme, they lower the activation energy of the reaction and increase the rate of the reaction. If the product is lower than the substrate, then the energy is released. And if the product is higher than the substrate, then the energy is absorbed. Activation energy is the energy which is required to start the reaction. That is known as the activation energy. So the enzyme function is to lower this activation energy so that the rate of reaction increases. And there are many factors which affect the enzyme. There's pH, temperature, substrate concentration, and the inhibitor. First, we are going to discuss pH and temperature. In pH and temperature, we obtain the bell-shaped curve. So here in this graph, the y-axis shows the enzyme activity and the x-axis shows the temperature. So at this point, this is the optimum temperature. At the optimum temperature, the activity of the enzyme is maximum. But if we decrease the temperature or increase the temperature, the activity of the enzyme decreases, because we know that most of the enzymes are protein. And as we decrease or increase the temperature, the denaturation of the protein occurs, which will affect the enzyme activity. Same graph we obtain in the pH also, that at the optimum pH, the enzyme shows the maximum activity. But if we increase or decrease the pH beyond the optimum range, then the activity of the enzyme decreases. Next, we are going to discuss is the substrate concentration. When we increase the substrate concentration, the activity of the enzyme increases or the rate of the reaction increases, but to a certain limit. After a certain limit, if we keep on increasing the concentration of substrate, the activity of the enzyme will not increase or the rate of the reaction will not increase, because at a certain limit, all the enzyme they get saturated. Now there's no availability of the free active site for the substrate to get bind. Okay, so initially the rate of the reaction will increase as the concentration of the substrate increases, but to a certain point. After a certain point, the rate of the reaction will not increase, because at that point all the enzymes they are occupied, because at that point all the enzymes are saturated and their active sites are already occupied by the substrate. So there's no availability of the free active site for the substrate. Next, we are going to discuss is the inhibitor. Inhibitor, they decrease the rate of the reaction. Malonate is the competitive inhibitor of succinate. Competitive inhibitor, as the name suggests, competition. So there's a competition is taking place between the substrate and the inhibitor for the active site of the enzyme, because in the competitive inhibitor, this malonate, they have the similar structure as the succinate, because they're having a similar structure as the substrate. So there's a competition occurred between the malonate and the succinate for the active site of the enzyme. And here the enzyme is succinic dehydrogenase. So malonate will act as an inhibitor for the enzyme succinic dehydrogenase. Next, we are going to discuss is the classification of enzyme. Number one is oxidoreductases. Oxidoreductases, they catalyze the oxidoreduction between the two substrates, and the example of oxidoreductase is peroxidase. Transferase, they catalyze the transfer of a group between a pair of substrate. Example of transferase are kinases. Next is hydrolases. It catalyzes the hydrolysis of ester, ether, peptides, and glycosidic bond. Example of hydrolases are peptidases. Next is lyases. They catalyze the removal of group from substrate by mechanism other than hydrolysis. Example of lyases are aldolases. Isomerase, they catalyze the interconversion of optical, geometric, or the positional isomers. Example of isomerases are epimerases. Next is ligases. They catalyze the linking together of two compounds. Example of ligases are carboxylases. So the enzyme is consists of two parts. There's apoenzyme and the cofactor. Apoenzyme is the protein part, and the cofactor is the non-protein part. And together, they constitute the holoenzyme, which is the biologically active enzyme. Cofactor, there are three types: the prosthetic group, the coenzymes, and the metal ions. Prosthetic groups are the organic compounds which are tightly bound to the apoenzymes. Example is heme group in peroxidase and catalase enzyme. Second is cofactor is a coenzyme. When the organic compound, they are temporarily bind to the apoenzyme. Example of coenzymes are NAD or NADP, which contain the vitamin niacin. Next cofactor is the metal ion. Metal ion, they show the coordination bond. They form the coordination bond with the side chain as the active site and with the substrate. Example of the metal ions is zinc, which is a cofactor for the proteolytic enzyme carboxypeptidase. Remember the cofactor, these are very important. Now let's discuss some of the questions. Question says that which of the following are not secondary metabolites in plants? I told you that secondary metabolites are the metabolites which are not essential for the normal physiological process. Options are 1 says morphine, codeine. Option 2 says amino acid, glucose. Option 3 says wind blasting, curcumin. And option 4 says rubber and gum. And the question is asking that which of the following are not secondary metabolites in plant. Remember, they're asking not. Morphine and codeine, they are alkaloid. An alkaloid is a secondary metabolite. Vinblastine and curcumin, they are drugs. Drugs are also secondary metabolites. Rubber and gum, they are polymeric substances. And polymeric substances are also secondary metabolites. But amino acids and sugar, they are primary metabolites. The example of primary metabolites. And primary metabolites are those which play an important role in the normal physiological process of the body. So the question is asking like which of the following are not secondary metabolites in plants? So the correct option is option number 2, that is amino acid and glucose. Next question says that which of the following biomolecule does have a phosphodiester bond? Option 1 is nucleic acid in a nucleotide. Option 2 is fatty acid in a diglyceride. Option 3 is monosaccharide in a polysaccharide. Option 4 is amino acid in a polypeptide. What do you think what will be the correct option? Which of the following biomolecule does have a phosphodiester bond? So the correct option is option number 1, because they're asking about the phosphodiester bond. And we know that the two nucleotides, they are joined by the phosphodiester bond, because the nucleotide, they consist of phosphate, the sugar, and the nitrogenous bases. Fatty acid in a diglyceride. Diglyceride, it consists of 2 fatty acids and 1 glycerol. And the bond here is the ester linkage. In polysaccharide, the bond is glycosidic. In polypeptide, the bond is peptide bond. And the question is asking about the phosphodiester bond. So the correct option is option number 1. Next question says match the following. Inhibitor of catalytic activity, B, possess peptide bonds, C, cell wall material in fungi, D, secondary metabolites. And here the ricin, malonate, chitin, and collagen. Choose the correct option from the following. You have to match the following. Those who are connected with YouTube, they can answer in the YouTube chat. Inhibitor of the catalytic activity is malonate. A2, possess peptide bond. I told you collagen is a protein. And the protein, they have the peptide bond. B, fourth. Cell wall material in fungi. Cell wall of fungi is made up of chitin. So C3. And D is secondary metabolite. Resin is a secondary metabolite. A2, B4, C3, and D1. So the correct option is 2. Now moving to next question. The essential chemical component of many coenzymes are. Options are vitamin, 2 protein, 3 nucleic acid, 4 carbohydrate. So the essential chemical component of many coenzymes are vitamins. Remember, the coenzyme, they are the cofactor. Remember, I told you there's a 3 type of cofactor. There's prosthetic group, metal ions, and coenzyme. So prosthetic group, they are organic compounds which are tightly bound to the apoenzyme. Coenzyme are the organic compounds which are loosely bound to the apoenzyme. And the example of coenzymes are NAD and NADP, which contain vitamin called niacin. And the question asking the essential chemical component of many coenzymes are vitamin. So the correct option is vitamin. Next question says, the primary protein are also called as polypeptide because. Option 1, they are linear chain. Option 2, they are polymers of peptide monomers. Option 3, successive amino acids are joined by peptide bond. Option 4, they can assume many conformation. Primary proteins are also called as polypeptide because. We know that the primary structure of protein, this shows the polypeptide chain, right? So the correct option would be 3, that successive amino acid, they are joined by the peptide bond. These are the amino acid, and these successive amino acids, they are joined by the peptide bond, and they form a polypeptide chain, which form the primary structure. So the correct option is 3. Next question says, in a reaction catalyzed by an enzyme, which of the following statement is correct? They're asking about the correct statement. Option 1 is enzyme make transition from substrate to product more difficult. Option 2 is the enzyme increase the activation energy for formation of transition state. Option 3 is the enzyme substrate complex formed during a reaction lasts for a very long time. Option 4 is the enzyme decrease the activation energy for formation of a transition state. And the question is asking about the correct statement. Option 1 says the enzyme make transition from substrate to product more difficult. No, the enzyme make the transition from substrate to product Easy, more easy, not difficult. Enzyme increase the activation energy for formation of transition state. No, I told you that enzyme, they lower or decrease the activation energy of the reaction, because of which it increases the rate of the reaction. So the enzyme, option 2 says the enzyme increase the activation energy for formation of transition state. No, it decreases the activation energy. Option 3 says the enzyme substrate complex formed during a reaction lasts for a very long time. No, the enzyme substrate complex formed during a reaction, it lasts for a very short time. Option 4 says the enzyme decrease the activation energy for formation of transition states. Yes, this is the correct statement, because the enzyme, they decrease or lower the activation energy for the formation of transition state, and hence they increase the rate of the reaction. So the correct option is option number 4. Thank you all. Thank you all, and see you in the next class.